concrete holds the, I used to watch the trees turn in Central Park from my office window in my one of my former jobs, and it was always later than outside the city because the concrete holds.
This is going to be so it's a pretty unique global community call, but it's going to be also pretty much informal. And the idea also is to get news from you after those couple of months of summer, but also to well, discuss about the future of OpenCovid19 and the opportunities we have, and how we're going to continue support and help each other, you know, in this crisis, because it's far from being over. So maybe let's start by giving you some updates from our side. So this is actually the 21st global community call. So we've been meeting a lot, and it's been basically more than 6 months, like exactly 6 months and a half that we started this community. So it's pretty amazing. And when you look at the results and the engagement, and it's really marvelous, and it's probably you know, a beautiful system to look at and even actually study and to replicate. So I'm really looking forward actually to see how we're able to sustain this program as long as COVID is still around. But it's definitely exciting. I think what we've done is a beautiful proof of concept that community-based science actually works. So I would say, like, congratulations to all of you. You made it happen. So regarding JOGL, so we've been doing a lot of work on the side to imagine how we could accommodate big communities like this one better. And one limitation, and several limitations we had in the past, and we still have, but we've been working on it, is that it's still difficult, for example, to like get recommendations, or know exactly when you arrive on the platform where you can start or how you can connect with someone or a need from a project, for example, stuff like that. So we've been working on a recommendation system for several months now, and with a team of scientists and engineers. There is also an open call where we discuss those matters, and so we expect to be able to release a new version of such a system by December. Full open source, as always, and that is going to be useful to look at and search basically the JOGL space to create connections. Another thing that we've been working on is a way for existing communities and groups of people to exist. You probably experienced it, but JOGL as a global community, as a way it is structured so far, meaning that when you join it, you become immediately diluted. It's like a giant sea. And so if you're, let's say, you know, like a community lab, or if you're an organization that has been working on several projects, or you're just actually another initiative, like there's been working on COVID-19, such as Corona Y or helpful engineering, it's hard to exist on JOGL. So we've been working on what we call so far is kind of like a nickname, but subspaces, kind of like a bubble within JOGL, where you have like complete control over your own community, and so you can, you know, basically manage the project within that community, the community itself. You can have your own changes and stuff like that. So that's going to transform quite a bit the experience you have on the platform, and so you'll be able actually to join communities by themselves, and enable organizations to exist also on JOGL. So it's going to, I believe, enable more meaningful also connections and in animation, and also sharing of resources. As for example, we've been talking with research institutes, and they're very interested in joining such initiatives. and being able to support also projects that they match with, even with funding. So that's, we're looking at an interesting future here, to support more independent and grassroots scientific and innovative projects. Well, so that's been the most things we've been working on. The other thing is we've been working also on other programs, but it's a bit unrelated to COVID. Regarding the following of what's the future of OpenCovid19, we've been discussing with our current partners, and especially the AXA Research Fund, which has been mostly behind us to create the microgrants. And the reason that we haven't yet launched another round is basically that we don't have the necessary funds actually to create such a microgrant round. But we've been working very hard actually to create such a new round, even actually several other ones. In fact, we're working to gather enough funds that we can sustain the whole program and the whole ecosystem by the end of 2021, because we believe that we need to get till the end of this crisis, and we hope it's going to be ended by the end of 2021. Probably not, but at least we'll have some more serious solutions by then. So in terms of microgrants, we expect to be able to create a new round of microgrants around, I would say, like in 3 to 4 weeks. That's the expectation. So unfortunately, we won't be able to do it before. And I know a lot of you have been expecting to get this new round. But yeah, it's been in discussion, and it took some while. So obviously, I will keep you updated, and in order to for you to apply correctly, and we've been also in contact with some of you, some of the projects, especially the one that just got into the X Prize ramp up pass, as they need extremely urgent assistance, and it's just a pretty unique opportunity, and they will be able to speak up about this crazy adventure themselves. They're here in order to be able to sustain them, so that they get the opportunity to participate until the end. All right, so that's pretty much everything on my side, except that we've been discussing about what's going to happen to community calls, and we are going to continue hosting community calls probably like once every 2 weeks, but in a different format. The idea is that at the end, we can still have a moment of discussion and sharing and open sharing as a community, but it feels like we need to have maybe like something more structured, and we're maybe a little bit more focused on a specific subject, so that you don't have to participate to all the calls, and you may be interested in one subject more than another. And so we've been thinking of doing two different things. One thing is creating an update for the global community, and so the idea will be asking projects who have results to share with us some results. We'll probably actually interview you like a couple of minutes through Zoom and record that, and we make like some kind of video editing, and we'll be releasing a video on social media so that people look at the updates of the different projects. And then the other thing that we will be doing is kind of like an evolution of the JOGL Live, where we focus on one project, and we enable the community to come around that project and discuss the changes that project have. One way of making this format evolve is to not only invite a project and the community, but also an external expert of that subject. Let's say we could, I was talking about the diagnostics. Maybe we could make a special session just about diagnostics and invite one or two external experts that could speak about the status of testing on the field, in different aspects, and discuss about with the project leads working on diagnostics tests together, and have something more of a thematic. And we could do that on the different thematics we have currently in the community, and create more meaningful content too. So that's the thing we've been working on. And in fact, if you have some remarks or ideas about how we could make those formats evolve, we are very much willing to hear from you, as this is also, we've been building those things for you, and so that it needs to be meaningful for yourself, basically. All right, so I cannot wait to hear from you guys. Who would like to start?
I mean, I can give an overview of the X Prize thing really quickly.
Sure, thank you, Ellen.
Yeah, so for anyone who hasn't been following this, actually, Chris Mason is a professor at Columbia University in New York, and he's actually on JOGL. He's been doing COVID-19 diagnostics using a technique called isothermal amplification. And so he told us all that there was going to be an X Prize around COVID-19 testing. And he urged us all to apply. And it turns out he's one of the consultants that they're using to devise this X Prize. And so we were able to, several teams from JOGL were able to apply. And it was, I won't go into the application procedure, but it was quite lengthy and a bit chaotic. But we all made it to the next round. And the thing is in several rounds. The first round is what they call the semifinals, where they send you a bunch of blinded samples to test, about 200 samples. The timeline of this is very aggressive. There was less than maybe 3 days between the time that the application deadline ended and the time they announced who would get to the semifinals. I believe they're going to send these samples out very soon, because they say that they will choose the winners of that round from people who successfully identify the samples by October 6th. And they will choose 20 out of that first round, which they said was about 200 people or teams. By October 6th, they will identify 20 that they are calling finalists. And those teams will have to deploy at least 500 tests per week to be done at clinical centers that the X Prize has partnered with. So basically, you have to send them your protocol and some sort of kit of your stuff. And then after that, they will choose 5 teams that will be basically their winners. And those teams are expected to ramp up even more, and they have a very ambitious but very poorly defined at this point goal of disseminating these tests widely across the world. So the semifinal round that we're going into, they provide no funding for the 200 teams who get these test plates. The final round, finalist round, where they've got 20 teams, they provide $50,000 per team to be spent on the testing supplies that they're sending out. And then the finalists, the ones that are the winners, the 5 winning teams will each get $1 million. And so we've got 4 teams in the running, which is great. And they can each speak to their own tests. And right now, we're all anxiously awaiting these test plates.
Thank you, Ellen. Yeah, this is very exciting. As you said, we're still actually waiting to see the real way is going to happen, those testing, when it's going to happen or not. Is there going to be any delays? Do you want to, it would be great for the ones that maybe didn't hear about your project before, yeah, how your projects differentiate from each other. Like maybe you could say in like in 1 minute or two, for the one that are here, what your projects, your detecting test rely on.
Okay, I guess I'll go first really quickly. We decided we wanted to use a commercially available kit that you could swab your mouth, and then the swab goes into an inactivation buffer so that the virus is inactivated, because we thought that was a very safe way of collecting samples. We also used isothermal amplification because you can get a result in 30 minutes that's colorimetric and you can see it with the naked eye. And then we had to find a bridge between those two sets of chemicals, basically, so that one would work smoothly with the other, and we found another commercially available substance. So we essentially strung three things together, and the test is 1 hour from the time you swab your mouth to the time you get an answer. And it requires no lab equipment except for a pipetter and some heat source, either a water bath. We used a sous vide cooker.
Thank you, Ellen. Guy, do you want to give a shot?
I can. So we also are. All the 4 teams are working on isothermal, so you don't need a thermocycler. It all works at one single temperature. And ours is based on fluorescence, basically. So we also have this little super cheap fluorescence detector, and that allows us to multiplex too. I mean, in the same tube, we do a test both for like a human gene, like an internal control, and also for the COVID, basically.
Awesome.
And then there's also Team Ali is also using something similar, but he has these different like waxes in the tube. So they make little cartridges and that melt at different temperatures. So we can have different reactions happening at different temperatures in different parts of this little tube.
Okay, thank you guys. So we're all very excited for you, and we'll all be supporting you in the next phase. Do you want to say anything, maybe Sarah or Martin, to complement what has been said?
No, I think I'm working with Guy, so I think he covered it all. There is the freeze-dried aspect.
That's true.
The cold chain independent shipping is another part of the corona detective
yeah, that's a very important part too. So we're able to freeze dry the reaction, so we can send them anywhere. And we're gonna send, there's also a team from Sri Lanka that is probably nobody on the call, but they also got through. And we're also gonna be sending out a bunch of kits to them to test there.
Awesome.
Yeah, I don't know, maybe not much to add from me too. I'm on the same team as Ellen and Chris Monaco, but I think she mentioned the main points.
Okay, thanks, Sarah. So let's move to the next project. And I see that, so I don't know if you saw, but there is a Google Doc that has been shared. I'm going to share it again, where you can put your project to be called, if you want to talk about it, just share it right now. So I don't see any, oh yes, so Catherine. Catherine, do you want to tell us more about the success of the project?
Yeah, so I finally have some updates, so I can share them with you. Yeah, my part of the open enzyme production project is open source enzyme optimization. And we finally agreed on the strategy. So we will go in with evolutionary information. Our goal is to make the enzyme more halo tolerant. Do you understand me actually?
It's okay. We can understand it.
All right. Yeah, I just have headphones without a microphone. Yeah, so we did the relaxation of the PDB file with Rosetta this month, and we got a grant for computation and CPU and GPU from the supercomputer at my university, and we got a sponsoring from Amazon Web Services as well. So we are good. We are set up with computational power, I hope. And yeah, I will implement the purification protocol, the updated from Ternan. It's on the protocol IO on the in the reclone group for the BSC long fragment. And I guess this is what I will use. So I'm still working on the real-time PCR assay with the wild type, so I can benchmark it, the mutant more than just with an endpoint, just the performance during the whole reaction. And yeah, this is still work in progress, but yeah, I finally got the commercial enzymes, and I also got the specific activity assay. So it's a phosphatase fluorescence assay from Biozyme. So thanks for that. Yeah, and we will do the first mutations, I guess, in 3 weeks on the in silico structure. So yeah, then after we find some stable or some good confirmations, we will do a MD simulation of 200 nanoseconds in GROMAC. So we somehow can assess a little bit better if it will be stable or not. And we can also go for osmolarity and pH. And if we found some in probably 2 to 3 months, I will do the the site-directed mutagenesis in the wet lab again and benchmark it with a real-time PCR assay that's still not functioning properly. So yeah, there's still a lot of work to do, but finally I could share some updates with you guys.
Thank you so much, Catherine. Yeah, those are great news. Don't hesitate to even actually make a small summary. You can post it on your feed on your project page on JOGL, so that people can react, and that would be fantastic. All right. The next project is the open enzyme production with Scott.
Hi there. Can you hear me okay?
Hi, Scott. Good to see you.
Good to see you too. Good to see everybody. So yes, the project's progressing. We're still waiting for the free genes to distribute the synthesis of the backbone vector parts to be able to assemble the fusion partners so that we can have like silicon purification or cellulose binding domain fusions to some of these enzymes to be able to purify. But I've been able to get the lyophilizer working, so I've been producing cellular reagents for these, which is kind of like the simplest approach to producing the enzymes. So it's been progressing. We've been impacted by the fires down in the south in the U.S. on the West Coast, up here in Vancouver. So it hasn't been pleasant in the labs, but
Wow.
Fortunately we've had, pardon?
Do you have red skies and the whole bit?
Well, kind of pinkish, but we've had rain overnight, and that's really helped a lot. I haven't been outdoors yet, but we've all had this constant burning feeling in our lungs. It's nothing compared to the people south of the border and the impact they're experiencing. But they rate the toxicity or the particulate matters on a scale of 1 to 10. Vancouver's above 10. or it was up until the rains. So it must be way worse down there.
Yeah.
But yeah, the project's moving forward.
Have you gotten the cellular reagents to work?
I don't actually
Ish.
have the primers, the positive control for the primer set. So actually, it'd be great if somebody here could send me some of the positive controls.
You have the primers, but not the end plasmid.
I've been actually waiting to order the primers because I want to know which primer set is the best to pick.
Okay.
But that would be quick. But yeah, no, I haven't tested it for the lamp assay yet.
Okay, but you at least in theory have like cells lyophilized that are expressing BST.
Yeah,
yeah. Cool.
Yeah.
Awesome.
Yeah.
Okay. So let's maybe send each other
sure,
back and forth some of that. Cool.
Yep.
Awesome.
Where is it easier to send from Europe or the United States?
It probably doesn't matter. Yeah, it's pretty much the same supply chain. I think things have improved a lot. I don't know about the U.S. Postal Service. It sounds like it's getting a bit of a hit.
Yeah, even before this, it was taking like 5 weeks. I got a couple of things sent from the Netherlands, from Jan Martin, and it's many weeks.
Yeah. No.
On the other hand, I got an overnight FedEx from.
I sent FedEx to Chile and New York and Sri Lanka, and it took a few days. Whenever.
Yeah, FedEx has been fine, but I think they've all kind of ramped up now. I had lots of problems with DHL in the earlier stages of the pandemic, and DHL also went wayward. So DHL isn't a big one in North America, but it's big in Europe and Asia.
All right. Thank you, Scott. Thank you very much. So I think we're done with the current project. We have some new projects we'd like to speak. So the first project I see on the list is the RADVAC, Open Source Vaccine Collaborative. It's Ranjan. Ranjan, are you here?
Yes, hello everyone.
Hi.
Hello and bonjour. Nice to meet everyone. I think in the future when we all have our neural implants, it'll be nice to hold the meeting in whatever language of the city it's being hosted in. So that'll be fun. We can wait for that in the future. Yes, the RADVAC project is an open source SARS-CoV-2 vaccine project based in the Boston, United States area. And there's a small group of us, 5, who got together to produce the formulation and to let everyone know about it, everyone in the open source community, the DIY bio community. And the objective is to essentially have a distributed research effort. We've started the effort on our side. We actually have 24 people here and climbing, actually more than 30 now, who have taken the vaccine. It's an intranasal administration. And we've taken saliva and blood samples, and we'll be testing those. So we've made everything available in the white paper that's on that website. And we would just invite people to take a look at it and let us know their impressions. Any questions that are asked are very helpful too, because it informs us as to what people are thinking and what maybe needs to be added to the white paper and the website. And we invite collaborations on multiple fronts. Some people will want to self-experiment. Some people will just want to work on the formulation. Others will, have the testing pieces, which we're also looking for because that's important for validation. So, yeah, we invite any communication.
So, just to understand precisely, so the project is what kind of experimentations do you perform currently?
Yeah, the whole idea was to sort of fast track vaccine development. And so we opted not to go for a commercial approach. We opted not to go for an approach that would fall under conventional regulations. And because of the self-experimentation approach, and self-experimentation is still legal in the United States, I don't know about other jurisdictions, we were able to create this formulation and self-administer. Each person in the group has chosen to give themselves the vaccine, make it and give it to themselves, and collect samples and contribute n's of 1 to a larger data set. So this is born of the realization that commercial vaccines are going to take some time. The world is in a situation now where lives are at stake, health is at stake. There's economic consequences, and so we're trying to accelerate the solution by doing it this way.
So I understand the pitch.
A solution, I should say.
Yeah. I understand the pitch. What are the identified limitations and risks that you got with this approach?
Yes, there are many, of course. The limitations, of course, are those in numbers. Statistically, having a much smaller cohort versus the commercial approaches, which have now thousands in phase 3 and had many hundreds in phase 2 and so on, is a limitation. The risks we've outlined on the website and the white paper, and we very clearly indicate this is a purely experimental vaccine. It's in fact an experimental approach to vaccine making, and we outline the risks, which is that there are very many unknowns, although the vaccine design is based on very well studied principles. Itself is an unknown, and so those are enumerated.
So an important part, can I just ask a question? An important part of the traditional vaccine development route is the informed consent.
Yeah.
That ensures that anyone who is volunteering for a study, and I know that there are problems with that system, but in terms of the spirit of that, you want to make sure that whoever is volunteering understands completely what they're getting into. And in this case, there's very little data. There's stuff that you pulled from the literature. There's a very small cohort. And right now, of course, most of the people that you're contacting are educated consumers, if you will. They're people like George Church. But if this becomes something that is disseminated more widely, do you have any mechanism? For example, when George did the PGP, which is a study where people get their DNA sequenced and the sequence becomes public, the informed consent was actually an exam. You had to read a lot of material and a lot of stuff on the history of human experimentation and what DNA was, and you had to then pass a test online to be in the study to prove that you understood the material that was given to you. Not just that you had been given material to read, but that you actually understood the content. Do you have any plans to develop anything like that, or are you just kind of releasing your protocol and the website to the public without following up on who exactly is taking advantage of that and trying to do it?
And those are great questions. We do have a terms of use informed consent currently on the site now, to which people have to agree before they can download the white paper. Of course, the white paper can be distributed in other ways, so the white paper itself contains those conditions at the beginning. We don't have plans to do an exam format, as you described, because that actually is actually fairly uncommon with informed consent, but we are working on a more extensive version. The tracking is tricky because it's an open source project, And so we don't enroll volunteers. We don't keep track of who has the information because we simply can't. It's a paper like any other scientific paper that just can float around, by multiple means. So that, because it's open source and because we are not a study, we are making this information available freely to stimulate in the scientific community, in scientific communities, I should say, further research using this vaccine platform and vaccine approach. It's not practical to, or even it's not doable to track consents, although we are working on a more descriptive consent document.
So you say that you know more than a paper that is circulating freely, but you're actively recruiting people for that study, right?
No, we're not an organized study per se. It's as I said, it's ends of one. It's individual investigators who each will review the approach, decide whether how they'd like to contribute to the approach if they want to at all, and then a number of us have decided to self-administer the vaccine.
But,
so there's
So I understand the science part where you try to combine current knowledge of vaccines development, but how you're going to prove that this vaccine that you're developing is working if you don't do a study?
Yeah. Oh, I see what you're saying. Yes, that's an excellent point. So this is not a self-contained approach. It is something that can work in concert with, in parallel with, conventional approaches, which have tremendous strengths, of course. So we are speaking with a couple of commercial organizations to take the study of the current vaccine formulation and future vaccine formulations, because it's updated constantly based on new research, to take those formulations and test them in a formal clinical trial setting. So that is definitely in the sights.
But so then you're planning to do to gather data regarding the people that are testing.
Absolutely, yeah. So what we have right now are, which we haven't released yet. We have preliminary data, but we're still working on the testing methods themselves because the vaccine formulation is atypical. Given that it's a large mix of peptides that address, multiple immune stimulation. So. But we have samples from that small cohort. So those samples will point, towards where the formulation and study should go ongoingly. But the open source approach and the release of the white paper now are motivated by the fact that the clock is ticking, on people's lives and health.
So I think I understand the pitch, but I feel like. So we've been seeing a lot of rushed science during this crisis, and that has harmed quite considerably not only the scientific community but also institutions that have we're surrounded by sometimes people claiming that this is a fact and when it has not been studied correctly or has been proven or been correctly reviewed, etc., etc., etc. And the hydroxychloroquine is one example, for example. How are you not such another example, for example? How are you not another hydroxychloroquine?
Yes. Great question. So for hydroxychloroquine, as an example, so there's a couple of sides to that. Of course, there's the political side, which was a total fiasco. And of course, we're not, luckily not subject to, usurpment by that particular group of individuals who would do that. The scientific part of it, we are we're very transparent. There's a reason that we haven't released data yet, though we have very preliminary data, is because it's not a data set. And we do not want any misinterpretation. So we make very clear that results are not available, that there are many unknowns, that this is purely an experiment and carries risks with it. So we are very careful to distinguish this scientific research from any conclusions that were asserted in other cases. So I would say it's in a different class, you know, as the example you brought up and other examples that we know of.
I guess one of the things that I would worry about, though, is that normally, and I understand that you're being transparent about the potential risks and the fact that nobody knows whether or not this is an effective treatment, but normally there are animal studies that are done beforehand to prove things like safety. And again, this is a flawed system. There's still things that would get by that. And maybe in nasal vaccines, it's more difficult. I know there are very few animals that you can use as models for things like flu. And they're pretty awful to work with, like cotton rats. But, you know, again, I keep coming back to this informed consumer aspect of it, you know? And the fact that you're essentially doing a phase 1 safety trial with volunteers without any institutional review board, without any screening, any other aspect that's being, go ahead, sorry.
Yeah, absolutely. So the way that we're conducting this is fairly incompatible with typical IRB studies because it is not a fixed design. The vaccine, for example, has gone through almost 9 iterations of design, and parameters are changing. Little parameters are changing, have been changing every 2 to 3 weeks. Again, an accelerated approach. So but you know, in terms of ethics, we have spoken with a couple of bioethicists, and their input has definitely informed the design and what we're doing. In terms of animal studies, again, we decided that given the extremely safe design previously demonstrated in animal and human studies of similar vaccine formulation and of the vaccine ingredients, that we were comfortable, a number of us, going ahead using ourselves as the test animals. And we did this before we released any information. So 30 plus people over a period of over 3 months now, over 3 months, in some cases 4 months, with multiple administrations between 3 and 12 doses now, with minimal side effects, stuffy noses, and mild headaches that resolved after 48 hours to 72 hours. That's been it. And so we recognized the importance of essentially doing a phase 1 ourselves to demonstrate that short-term safety. And short-term safety, of course, is the only thing that can be, you know, claimed even in longer-term studies. You know, a year is still short-term. So we've done that, and you know, sort of taken that responsibility first. So now that we feel comfortable, we're, you know, releasing the information, and we have released the information.
I think this is a fascinating discussion, and it's a provoking one too. So currently, if I will have to wrap up the discussion because there are other projects on the line waiting. But to give you a fast feedback, so in your current format, so we've been working with the community on precise biosafety and biosecurity guidelines. And with the current format, like, you don't fit within. And so in that aspect, we cannot like support you like formally as we've done with other projects. However, I mean, the idea of doing collaborative development of a vaccine is not wrong, and an open source one. And it feels like there are such good, scientific and safety protocols out there that it would be a waste to just remove them just for the sake of, speed. So if you're interested, I am to take this discussion further another time, maybe have a special call for that, and we can discuss about how we could support such initiative at the conditions of maybe changes, in the protocols. But because I mean, it's there are some good parts in it, definitely, and it would be a waste to throw it away.
That's I really appreciate that, and Ellen, I really appreciate your questions as well. Thank you for so much time.
I have to say, I've been really tempted to try it. I've been very tempted to try it, but the safety aspect has held me back.
Yes, yes, of course. That gave me pause a number of times, but with careful reading and rereading of of our work, and thinking about it, repeatedly, it was sort of, in my mind, scientifically sound, and I was also, and we all have altruistic motivations, and so that factored into my decision as well. But thank you so much for all this time. I wasn't intending to take so much time, and we welcome off the JOGL platform any inquiries, of course, at the website, and Thomas would love to continue discussing how this could be adapted, because the name of this, the middle name of this project is adaptive. So
organize a call, around maybe next week. Are you on the JOGL Slack maybe?
Yes, just joined.
Okay, perfect. I'll send you a message so we can set up something.
Okay, thank you.
All right, thank you. So let's move forward. So we have then a new project, Adam, with Nuclid, which is a no pipetting special equipment lamp test.
Hi everybody, my name's Adam. I'm part of the team Nuclid. We're a small team based in Singapore, so we're a combination of mechanical designers and microbiologists. What we're doing is we're developing a COVID-19 test where our main objective is to remove a lot of the procedures that are involved in a traditional PCR test, so the pipetting and the transfer of liquids from different tubes to the other tube. So we're using the NEB isothermal lamp kit. But what we're doing is we've developed a small little cartridge. It's about the size of a pack of cigarettes, which you can put the patient sample in, so you can spit into it, you can close it. You can put that on a heating block and inactivate it. But then inside the device itself, it's non-electrical, it's purely mechanical, but then that will transfer the patient's sample, perform the RNA extraction, and then move that template in a precise amount into the PCR reaction. So in summary, it's just that we want to create a very simple, low-cost, easy test that really anybody can use anywhere.
And
say a few words. What kind of extraction you mean?
So we have two ones we're looking at. So one is we're using a column filter. So just like the little kind of fabric filters. I don't know what they're made of, but and then the other one is we're using silica beads. So we have two different kind of parallel designs we're working on.
Cool, and it's a cigarette pack size device, the whole thing?
Yeah, yeah.
Wow.
So when you say mechanical, I'm picturing like dominoes, like like somebody's hitting something else and is going. What do you mean by this?
Yeah. Do you like press a button?
Yeah. So sorry, it's really a very kind of simple microfluidics device. So there's like some plungers that you kind of move, and it's almost just like kind of an exaggerated syringe system, if you like. And by moving the plungers, and we designed the channels in the way that they move the liquids in precise amounts. So we can take 5 microliters of the template, and then we can put it into the PCR tube after we've done the RNA extraction. So it's like a small little neat cartridge that you can just spit into, seal it up, and then you like have a plunger which you can move up and down, and then that'll like pump the liquids through the different stages.
That's great.
So do you have a picture or a video?
I was going to ask.
Yeah, I can. If you give me one, I'm just in a different room. If you can bear with me like 1 second, I can.
Sure.
Honestly, I can imagine somebody's like, oh, result. And how long does it take, did he say?
I don't think so, no, he didn't.
I mean, it does lamp, but that's it. And the extraction doesn't usually take a long time. And I think they're still playing around with the extraction. They haven't checked how well the different ways work.
I wonder how it's powered.
No, it's manual. That's what he said. It's not electronic. With your finger, you like press a syringe and that moves all the stuff kind of.
But do you not need
And then you just, well, they lay it, they said they lay it on a heat source.
Yeah, you
just
put it
add
on
heat
a
block.
like heating block, but you can also just make a heating block with like resistors basically.
So we have two, this is like the first one. So this is using the column filter. So it's a little bit broken at the moment, but like this would be your sample container. So you can put your sample in here. This has a little lid, and then you put it in here. And then inside there is a column filter inside, and you'd pump this. And then in the bottom there is a PCR tube, and then you can just put that in your heating block. So after the template is moved through the column filter, it goes into a waste chamber, and then there's an elution buffer that you put in as well. So you can take this out, and then you put another one in which will have the elution buffer, and then that'll force it through the filter into the PCR tube. It's a bit more, it's quite complicated, that one. We have this other one, which is a bit simpler. So it has 2 parts. So you have this, which is just like, it uses like a Coca-Cola bottle. So you can like spit into it, and then you can seal this up. And then this will just fit in a standard heating block. So you can just inactivate this. And then we have a lysis buffer in here as well. And there's a kind of special seal that will break when I screw it into this device. So as I screw it into this device, then the seal breaks. And then that allows the sample from this container to move into the rest of the cartridge. And then you just kind of pull these little plungers up and down, and then that'll move the liquid into these little PCR tubes. And then you just drop that in your heating block. And it's just isothermal lamp, so it's a colorimetric reaction, and then you just take it out and you can see the color afterwards.
How do you do your controls?
So this one, so you can see it has two tests. So there's one control. So we're using an acting control in one of them, and the other one is the real test.
Very cool.
Do the tubes need to stay in there, or does it?
So this is like the really big challenge we have is we will have them taking off because the shipping is really difficult because we're based in Singapore. So to cold ship the PCR mix is just a nightmare. So yeah, we have the tubes actually removable, and they go in your cold shipping, and then the rest of this bulky kind of plastic unit just goes in your normal ambient temperature shipping. So yeah, these tubes they're not on this one at the moment, but they are removable, so you can just pop them on.
What is the cost?
I'm also worried about evaporation if you heat it when they're still connected.
So before we get into too many details, you guys hot it up.
I still have questions.
We can take it offline.
Yeah, exactly.
You can I also buy the vape cartridge to plug in there? Like,
Oh, right, yeah. I think it's
That's
inspirational
cool.
to my spent youth. I don't know.
So a couple of questions very fast. So is this, so the whole thing that you've created is open source, or are you going to planning to post the documentation at some point, or what's your plan basically?
At the moment, we're just building this. I mean, we are a small startup, so yeah. I mean, we have no revenue at the moment or anything, but I mean, we're here. I mean, we're sharing the design, and yeah, if people want to collaborate or help, yeah, it's
because so that's that you probably have seen. That's the principle of this community and the platform, that it's not about who's going to succeed. It's really about building something that works and that we can share afterwards. And so yeah, so I think this is great. The community has many questions. What would be great is if you can share some more info, on your JOGL profile, so that people can see it. That would be fantastic. And otherwise, yeah, we have a special Slack channel for the diagnostics. It's called nucleic acid amplification. And so if you're not on it yet, I encourage you to join it, because that's where everything happens.
Yeah, he is.
Yeah, I'm on the Slack.
Perfect.
I have got my project's not very up to date, but it is on JOGL. There are some like pictures of this one you can see on there.
Awesome. So yeah, so it's great. Do you want to ask for anything? What are your current needs basically?
I suppose one need, we are looking for like lyophilized tests. That's because, like I mentioned, the shipping is like a really big challenge for us, because the cold ship, like the box is like this big, and then you can only put this much stuff in it because the rest is just dry ice. That's one challenge we have. But I think at the moment,
I think one of the next steps for Sarah is, NEB says that they haven't had success in lyophilizing the colorimetric assay, but that they think it could be done if someone worked on it.
Yeah, we had that.
So we would certainly share any information if we start doing that with you.
Okay, cool. Yeah, I heard it was because of the glycerol or the glycol that's in the...
Yeah, it's the glycerol, but then it's also the pH. Something goes wrong with the pH with the glycerol free and stuff, supposedly.
All right. I'm glad
because they sell the individual reagents without the glycerol, but they were never able. They could do it with the fluorescent, but not the colorimetric. You should look at Guy's project, definitely.
Yeah, I've seen Guy's project. Yeah, that's the other one. Yeah, I know the fluorescent one can be lyophilized. Yeah.
So I think I'm super glad you guys connected. Obviously, you have many things to talk to each other, to tell each other. I'm going to move on to the next project, which is the Cosmic Medical and the OxyKit. Do we have someone here to talk about this project? I think Yajour. Yajour, are you here?
Yeah.
Hi. Could you tell us more about this project?
Sure, yeah. So I'm currently support a group called Cosmic Medical. They're a non-for-profit startup that was launched at the University of British Columbia. They're creating a variety of designs for very low-cost ventilators, oxygen concentrator systems, and basically everything that moves between those two. And so right now they're also collaborating with OxyKit, who are producing a open source oxygen concentrator. Because right now we've reached a saturation point for like commercial ventilators. So there's more than enough to go around. But now the issue is actually finding sources of that oxygen on site in location in a variety of places, such as like field hospitals in Afghanistan, rural hospitals in Brazil. And I think there's one more group or team they're also talking to in Argentina, but just a variety of different use cases and need cases for oxygen. And right now they're trying to increase the number of people who have engineering experience to support the project. So if any of you happen to know anyone who'd be interested or have that experience, they would love a few extra people to support that.
Cool. Have you shared those needs, for example, on your JOGL project page?
I will double check on that.
Okay.
Do you have kind of design specs on your available on the web or on the JOGL page?
So they've been hosting their designs on GitHub. So is there a way to like sync up the GitHub to the JOGL page, or would you just post a link there?
A link is fine.
A link.
Okay.
Post it in this chat as well, maybe.
Okay. Yeah, if you have any more questions or would like to connect with them, feel free to just message me on the OpenCovid19 Slack, and then I'll make sure that like someone can contact you like really soon from that team. And yeah, if you have any other questions, feel free to send them to me. And yeah, thank you.
Well, thank you, Jeju. And yeah, don't hesitate. Could you share the link to your JOGL page on the chat so that people can check it out? Jeju?
Oh yeah, I'm just like looking for it.
Sorry, I was not sure that you heard me. Perfect. All right, I think we're perfect. Awesome. So I think that we are pretty much done. So we had originally, we are planning to also present some work we've been doing regarding the methodologies and the processes that we've put in place during OpenCovid19 that we're summarizing into an Open Community Playbook. Alex had to go, unfortunately, and so the idea is that to not make this call too long, we'll present actually this community playbook in the next call. But you can actually have a look to it. The link is in the Google Agenda. It's a pretty cool collection of guidelines basically and ideas about how to animate and create an open community for science. And we're launching collaboration with other communities such as CoronaY and also potentially the helpful engineering community, so that we can actually bring together all those good principles so that it can be published openly afterwards. Otherwise, that's pretty much it. Are you ready for the traditional group picture? Let's activate our video stream, and I'll make awesome. I'll make a screen capture. Do you think that? Oh yes, fantastic. Nico or Manavi, can you activate your video stream? Oh, fantastic.
As you driving, Ellen.
Yeah. All right.
I'm parked.
Say hi to everyone. Fantastic. Thank you so much for this great call and great discussion. Very exciting. And I look forward to seeing all of you back on the Slack and the JOGL platform, but also for our next community call that will happen in 2 weeks, which we'll be sharing the details with you. But again, have a great day, great night, great morning, and see you all very soon. Bye-bye.